Hello everyone, I'm very excited to welcome you to my YouTube channel, Tony's Insight. So in today's video, we'll be talking on lipid metabolism. Our focus will be on how fatty acids are oxidized, or let's say how fatty acids are broken down to form acetyl-CoA. The reason that this pathway is so important, might be like, why is this pathway so important to us? Why this pathway is so important because of its used energy. It is the central energy yielding pathway in many organisms and tissues. For instance, in your heart and liver, it provides like about 80% of your energetic needs. Understand? So this pathway is very important. So I want to now discuss, because this fatty acids, the breakdown of this fatty acid, it happens in the cell. So I want to now talk about the sources of this fatty acid. How did this fatty acid get into the cell? So there are various sources of this fatty acid. Number one source, the major source, which make up the topic of the first part of this video, is the fat you ingest in your diet. The fat ingested in your diet is the first source. Let me go back again. I was saying that fatty acids are oxidized to form acetyl-CoA in the cells, and it's so important because of its energy yield. It uses a lot of energy. It wants 80% of the energy you need in your liver, in your heart, and many organisms. So now, talking about the function, where the sources of these fatty acids are, one is the fat you ingested. Let me call it dietary lipids. The lipids you ingest in your diet. So because this lipid you ingest in your diet will be digested and absorbed. From absorption, it will enter the circulation, and from circulation, it will then enter into the cells to undergo this positivity pathway. Understand? But the other sources, like the fats that are stored in adipose tissue, fats stored in adipose tissue. That's what cause fatty acids. Yeah, fats stored in adipose tissue. Other sources are from when lipids are recycled being autophagy. This one happens during starvation, or maybe lipids can also be synthesized from excess carbohydrates in liver. So these are the sources of fatty acids. Now, fatty acids, they include, what really are fatty acids? Or let me say better, what really is lipid? So lipids includes a variety of chemical substances. Lipids includes a variety of chemical substances. One is glycerol. This forms like about 90% of our fat intake. Because as an adult, we take about 60 to 150 grams of lipids per day. But this glycerol forms about 90% of it. Then the others, there is the fatty acids and its derivative. There's also the phospholipids. There is the glycolipid. The others, there is the vitamins, that's the vitamins A, D, E, and K. Then there's also the cholesterol, the sterols. So these are chemical substances that are found in lipids. I told that glycerol makes up about 90% of it. Others make up the rest. Understand? So so the topic of this video, really, this first part of the video, is we are talking about digestion and absorption of lipids. How do the fats you ingest in your diet, how is it digested, and how is it absorbed, and then how does it get into the cells? So, there's no absorption of lipids. So let's say you take a meal containing fats. No digestion really happens in there because it will pass through the mouth, through the esophagus, then to the stomach, and then to the small intestine. Digestion doesn't really, little or no digestion happens in the mouth and in the stomach. Where digestion really happens is in the small intestine, the duodenum of the small intestine. That's where digestion happens. So see what happens here. The general picture, one general picture of lipids is that they are insoluble in water. This insolubility. So lipids are generally insoluble in water. But the fact that they are insoluble in water, it presents a problem. Because inside the small intestine, it has to be acted upon by water-soluble enzymes. You understand? But they themselves, they are insoluble. So, but they have to be acted upon by water-soluble enzymes. So, what makes them able to be acted upon by these enzymes are bile salts. So, bile salts act on this lipid to kind of solubilize it, or the better term is emulsification. Emulsification. So these bile salts, they act to emulsify the fats. So these bile salts, they are produced in the liver from cholesterol. So when they are produced, they then get stored in the gallbladder. So it is in that gallbladder that it remains there. So you now take a meal containing fat. Once the meal gets to the small intestine, the gallbladder will release this bile salt, which will then act on the lipids, breaking it down. And that is the breaking down we want to talk about now. Not breaking it down, it act on it to emulsify it, forming mixed micelles. It will form what? Mixed micelles. Let me recap. I said that little or no digestion happens in the stomach. Little or no digestion happens in the mouth, or the stomach. Little or no digestion happens there. But where digestion really happens is in the duodenum of the small intestine. And I said again that initially, the lipids that got into the small intestine, they are macroscopic, they are big, and they are insoluble. But they have to be acted upon by water-soluble enzymes. They have to be acted upon by water-soluble enzymes. So that's why they have to be emulsified by what? Bile salts. This emulsification by bile salts converts it into finely dispersed mixed micelle. Okay, these are chemical substances found in lipids. So I can see that the step 1 in digestion and absorption of lipid is what? I can see that step 1 is micelle formation. Step 1 is what? Micelle formation. Whereby the bile salt will emulsify the fats in the small intestine. It will now make it microscopic and finely dispersed. Understand? The point of this micelle formation is so that it will increase the lipid function that can be acted upon by these water-soluble enzymes. So yeah, as it has now formed these mixed micelles, that's when digestion can occur. Digestion occurs by the action of what? Pancreatic enzymes secreted by the pancreas. So the second step I would say is, let me say, should I say hydrolysis? Yeah, hydrolysis by pancreatic enzymes. So that's the second thing that happens, hydrolysis by pancreatic enzymes. So let's take for instance, triacylglycerol that has this has chemical. So this is the chemical structure of triacylglycerol here. So this is the chemical formula. So this is what? Triacylglycerol. This is chemical formula. So when this triacylglycerol, when emulsified triacylglycerol, when it's then acted upon by pancreatic enzymes, specifically the lipids, you know what happens is that? Okay, what happens is that? That's when it's acted upon, that's when it's hydrolyzed by pancreatic lipids. What happens is that there will be break at this carbon 1 and carbon 3. So there will be something like this now. So what do you think has happened? This one now will go as free fatty acid. This one will go as free fatty acid. Remaining this one. So what we now form is 2 monoacyl glycerol plus 2 molecules of free fatty acids. Yes. So okay. We're going to write it. CH2OH here. Now CH2. So let me call this R1 out there. So this one now, CH2O. Plus what? 2 molecules of free fatty acids. Let me say, let me write this. R1COO plus R2COOH. So I want you to understand what happened here. Pancreatic lipases that was secreted from the pancreas came and acted on this. It hydrolyzed it. That's the action of H2. It hydrolyzed it. So see what happened here. There will be break at these 2 points. Carbon 1 and carbon 2 and carbon 3. There will be break. So CH2 now, this is the CH2. CH2, this is CH2 here. CH2, this is CH2 here. Sorry, this is OH here. Yeah. So see what happened. OH from here entered here. OH. You know, this is 2 molecules of H2. So that's OH and H. So OH here went here. Another molecule of OH here went here. You understand? Remaining, this is, so this part here now is 2 monoacyl glycerol. Yes. So, okay. So what have I said so far? I've said that when the parts get into the small intestine, the duodenum small intestine, they are emulsified. The point of this emulsification, is so that what happens? So that it will form micelle formation. Because initially the parts, they are insoluble, they are macroscopic. But when bile salts come and emulsify these parts, they become finally dispersed. They become able to be acted upon. They become microscopic and able to be acted upon by lipases that is secreted by the pancreas. You understand? So this lipase is secreted by the pancreas now. Within act not these enzymes. So assuming that it was glycerol, if it was glycerol, like I showed you, what will it form? It will form 2 monoacyl glycerol plus 2 molecules of fatty acids. So this is lipase acting here. The enzyme here is lipase secreted from by the pancreas. So if it were, let's say, cholesterol ester, what happens is that cholesterol esterase, the enzyme called cholesterol esterase will then come to break this down into cholesterol plus free fatty acid. If it were glycerophospholipid, let's say glycerophospholipid, it will be broken down by phospholipase A2 to form what? To form lysophospholipid. Phospholipid plus free fatty acid. Yes. So all these things right now are the primary products of lipid digestion. All these now plus bile salts, they are the mixed. That's what we call the mixed micelles. I'm saying that these lipids plus the bile salt, that's what forms the mixed micelles. Yes. So at this stage now, it is ready to be absorbed into the intestinal wall. Understand? So what happens? This mixed micelle will then approach the epithelium lining the intestinal surface. The mixed micelle will, because it is in intestine, right? This mixed micelle will then go and approach the epithelium lining the intestinal surface, the intestinal mucosa, to approach it, and then the lipid component of this, that's all this, will diffuse into the intestinal surface, into the intestinal mucosa rather. I'm saying that the lipid, what's the leading portion of it, that's all this, will now be absorbed, it will diffuse into the intestinal mucosa. So inside the intestinal mucosa, that's when another interesting thing happens. So far, okay, I'm not doing quite my steps. I said that the first step is what? The first step is micelle formation, micelle formation. The second step is hydrolysis. by pancreatic enzymes. It is in this second step that the products of lipids, the primary products of lipids are formed, which can then be absorbed to the intestinal wall. So the third one is just diffusion through into the intestinal mucosa. Where I just say, and just write it absorption. Yeah, absorption. Absorption. So the fourth one now is reconversion. Do you remember I told you that the triglycerides will be broken down into what? The two monoacyl glycerol plus two molecules of free fatty acids. The phospholipids, the glycerophospholipids will break down into lysophospholipid and free fatty acid. The cholesterol ester will break down to cholesterol and oil. So the fourth thing that will happen here is now the conversion. After it has been the conversion into the conversion. So after it has now been absorbed into the intestinal mucosa, it will then reconvert. Like the two monoacyl glycerol will combine with free fatty acids to reform what the triglycerol. The same thing, the cholesterol ester will be reformed, the phospholipids will reform, everything will just reform in the intestinal mucosa. You understand? Everything will form in the intestinal mucosa. So let me call this reconversion. You understand? When it's now reconverted, that's when it will then combine with apolipoprotein. So the third stage is combination. It will then combine with apolipoprotein. One call is combination with apolipoprotein to form lipoprotein. Okay? So what is this apolipoprotein? This apolipoprotein, they are let me say it, they are proteins that they are transport proteins. They are proteins that combine with lipids to transport them into circulation, right? So this apple means that when this apple means that it is detached, when it is free, when it's only the protein, but when it combines with lipid, so combining lipid combination with apolipoprotein to form just lipoprotein. So this apple means that it is detached, it is separate from any lipid. But when it combines with the lipids that is formed from the conversion of this, when it combines with lipid, they form lipoprotein. So, and when the lipoprotein aggregates, different molecules of lipoprotein, when it aggregates, it forms chylomicrons. So this one is like lipoprotein. Proteins aggregates to form chylomicrons. So in this form, this chylomicron forms, that's the form whereby it can then be transported into circulation. So let me just do a recap of what I've said so far. I've said so far is that lipids, that they include a wide variety of substances, chemical substances. We have the phospholipids, the glycolipids, the triglycerides that makes up 90% of your daily fat intake. Understand? So all these are chemical substances. They are lipids. Understand? So I'm not talking about what exactly happens when you ingest a meal containing these lipids, these fats. What happens? What happens is that it will go through your mouth, through your esophagus, and then your mouth, in your stomach. That's where it forms kind that goes into your intestine. So what I'm trying to say is that later on digestion happens in the stomach and in your mouth. But when it gets into the small intestine, that's where digestion happens. You understand? So when at that point when it went into your intestine, it was insoluble, it was macroscopic, and it has to be acted upon by water-soluble enzymes from the pancreas. Water-soluble enzymes from the pancreas. That's why you have to undergo change form. It has to change form. It has to become microscopic, finally dispersed, so it can be acted upon by these enzymes. You understand? So it's now bile salts that are secreted from liver with by cholesterol, like it uses cholesterol to synthesize bile salts in the liver, and it is called in the gallbladder. That is when this gallbladder then secretes the bile salt into the small intestine, it will then act on triglycerides, act on the phospholipids, act on it to digest it. Understand? So when it is digested, it will see what when digest it. Sorry, it will act on them to emulsify. So after it has emulsified, it cannot be acted upon by the lipids. not like this by the pancreatic enzyme, which include like this is the phospholipase A2, and so on. Understand? So the enzyme that is secreted by the pancreas will then come and act on it, breaking it down. Understand? It will break it down. It will break it down into a form that can be absorbed. So that was hydrolysis by pancreatic enzyme. Then absorption, when it approaches the flush border membrane, the epithelium that is lining the intestinal wall, it will approach it, it will then diffuse into the intestinal mucosa. Understand? So that was absorption. So after it has absorbed, what happens is that it will be converted back. The lysophospholipid will convert back into the glycerol-phospholipids. The two-monoacid glycerol plus free fatty acids, it will convert back to glycerol. Understand? So after it has been converted back, it can then combine with apolipoprotein to form lipoprotein. I told that apo means that it is separate, it is detached. But when it combines with lipids, it will then become just lipoproteins. So when lipoproteins aggregate, they form chylomicrons. Chylomicrons are the transport vesicle where they can then move into circulation. So when chylomicrons are then formed, next thing that happens is that they enter circulation. They first of all enter into the lymph, and from the lymph, they will then enter through the thoracic duct. They will enter into blood circulation. So it is in this blood circulation now that I want to talk about, because when you enter blood circulation, it will then go to the muscle. It will go to the muscle. It also wants to maybe go to adipose tissues. Understand? So it will go to the muscle, go to adipose tissue. So in the capillaries of this tissue, remember that I said that lipoproteins, the chylomicrons, they contain apolipoprotein. Understand? So the seventh step now is that apolipoprotein, right, that is contained within this chylomicron, it will stimulate another class of enzyme called lipoprotein lipases. What this one does is that it will then break down, for instance, it will break down the glycerol to fatty acids and glycerol. Yeah. So it will stimulate lipoprotein lipase to break down like that lipoprotein to break it down into fatty acids and glycerol. Yeah. So when fatty acids now get close to the plasma membrane, there are specific transporters in the plasma membrane that can then transport it inside the cell. It is now inside this cell that beta oxidation can occur. Understand? So you can go. I thought that when it gets into blood circulation, you can go to the muscle where it will enter the cell and the oxides to provide energy, or it can start to go to the adipose tissue where it will be esterified for storage. Because triacid rest will actually make good storage full. So when they are needed again, that's when they will then go and be oxidized to provide energy. Understand? So at this point now, where is fatty acid? So where is it that the lipoproteins that they are like the triacid rest, they are now broken down into fatty acids and glycerol. The fatty acids are what's absorbed. They are taken by the transporters in the plasma membrane. They are taken inside the cell. That's where they are being used for oxidation to provide energy. The first thing I said, the fatty acids are oxidized to provide acidic weight. So that makes, with that, we can see that we've completed the first part of this video. Second part, we're now looking at the oxidation of that fatty acids. If you have any question, remember to drop it in your comment box. If you have any suggestions, remember to drop in your comment box. Yeah. So please remember to subscribe, like, and share. Alright, remember to on your notification button so that anytime I drop a video, you'll be the first to get it. Alright, thank you.